Hey everyone, welcome back to the channel. Today we're going to talk about the neuromuscular junction and we're going to talk about the basic function of it, and then once we understand the function, we'll go over the 4 different ways that you can produce a neuromuscular block. And this is a question that I've come across in exam practice that I really struggled to answer because my brain instantly goes towards depolarizing and non-depolarising neuromuscular blockers, but there's actually 4 ways that you can produce a neuromuscular block. And I'm going to break this down and we're going to go over each of them, and it'll help us understand those niche areas of knowledge that will help us mop up some extra marks in the exams, and especially in the vivas if they do come up. So to begin with, let's talk about the neuromuscular junction. So the neuromuscular junction is a specialized synapse between the motor neuron and a skeletal muscle fiber designed for rapid, reliable transmission of a signal from the nerve to the muscle. You can think of it as having 3 main components. It has a pre-synaptic terminal, which is the nerve ending. It has the synaptic cleft, which is the space in between, and the postsynaptic membrane, or the motor end plate of the muscle fiber. Let's first talk about the presynaptic terminal. So this is the end of the motor neuron, or axon, and it's the sender of the signal to the junction. The key features here is that there's the synaptic vesicles, which are small membrane-bound sacs containing acetylcholine, and there's about 10,000 molecules per vesicle. They're clustered near the active zone. The active zone is the specialized region of the presynaptic membrane where the vesicles fuse to release acetylcholine, and these contain voltage-gated calcium channels, though the voltage-gated calcium channels are also found around the nerve end plate as well. They're opened by the action potential reaching the nerve, which is how that signal from your brain goes into your spinal cord all the way through the nerve to the muscle motor end plate, leading to the contraction. There's lots of mitochondria that you'll see in this area, and that's because you need a lot of ATP for acetylcholine synthesis and vesicle recycling. And this is because glucose is turned to pyruvate, then to acetyl-CoA, which is a substrate that is then combined with choline to make acetylcholine, and this is with the enzyme choline acetyltransferase. There's also high affinity choline transporters around this area as well, and they bring choline back into the terminal to be recycled. Then going in a bit more detail, there's these proteins called snare proteins, and this is things like synaptobrevin and SNAP25 and syntaxin, and they mediate vesicle docking and fusion. So you can think of them as these proteins that sort of attach to the vesicles and near the wall, and when they're activated, they'll quickly pull the vesicles to exocytose all of that acetylcholine. And then in the synaptic cleft, which is the second part here, this is a tiny extracellular gap. It's about 50 nanometers wide between the nerve terminal and the muscle membrane. But it's not just an empty space, it's actually packed with a lot of different structures. So it's not always drawn, but there's a basal lamina that sits here, and it's specialized extracellular matrix that holds everything together and helps align the presynaptic and postsynaptic membranes. Then there's the important enzyme acetylcholinesterase. So this enzyme is anchored within this basal lamina, and it rapidly hydrolyzes acetylcholine into acetate and choline, which helps to terminate the signal. It's important that it sits within this membrane here because it prevents it from diffusing away from the space, creating a high concentration of the enzyme in the space. And then there's the space itself, so the diffusion space, and the cleft allows acetylcholine to diffuse quickly to its transporters, but also ensures it doesn't spread too far, maintaining precise local control. Finally, we have the postsynaptic membrane, or the motor end plate. And this is the part of the muscle fiber membrane directly opposite the nerve terminal, and it's the receiver of the signals. So the key features here are junctional folds. So you can see that there's these deep infoldings of the sarcolemma that increase surface area and bring the voltage-gated sodium channels close to the acetylcholine receptors. The tops of the folds contain nicotinic acetylcholine receptors, and the bottoms contain the sodium channels for propagating the action potential. And this is important because if you think about it, if you've got limited space, you would rather have the receptors close to this where the acetylcholine is being released, and the channels can be a little bit further down as they activate and propagate to produce the action potential. Now, there's the nicotinic acetylcholine receptors, and they're a pentameric ligand-gated ion channel. In adults, they're made of 2 alpha, 1 beta, 1 delta, and 1 epsilon subprotein. While in infants and neonates or fetal receptors, the gamma replaces the epsilon. So each receptor binds two acetylcholine molecules, opening the channel to allow sodium influx and potassium efflux, so they're non-specific for the ion, and they produce the end plate potential. The binding of one acetylcholine leads to the increased affinity of another acetylcholine binding site through conformational change, and this is similar to what you see with hemoglobin and conformational change with oxygen. Then deeper down into these clefts is the voltage-gated sodium channels, and they're found deeper in the folds, and they amplify the depolarization to generate a muscle action potential, and there's also more acetylcholinesterase that sits down here as well. There's also the cytoskeleton anchors, which are proteins like rapsin, which anchor nicotinic acetylcholine receptors in place, ensuring that they remain densely packed. And there's about 10,000 nicotinic receptors per micrometer squared. So I'll briefly touch on this, but it's not always shown in diagrams, but there are several supporting structures. And these are things like Schwann cells, which cap the nerve terminals to help insulate and maintain the junction. And after injury, they play a major role in guiding regeneration. There's also perisynaptic fibroblasts. They help maintain the basal lamina and synaptic organization. And then there's synaptic vesicle recycling machinery that endocytose and recycle pathways, ensuring the vesicles are replenished after release. And these are things like the receptors that take in the choline that's broken down from the acetylcholine to help recycle that, while the acetate is diffused away. So we understand the structure now and the anatomy of the neuromuscular junction. So we'll talk about how it works. So the first thing that happens is that you have depolarization of the nerve terminal, which opens these voltage-gated calcium channels, and calcium rushes in from the extracellular space. That influx of calcium triggers vesicles containing the acetylcholine to move towards and fuse with the presynaptic membrane. And remember, they're half bound by those snare proteins. The acetylcholine is released by exocytosis into the synaptic cleft. It then diffuses across, binds to nicotinic acetylcholine receptors on the motor end plate of the muscle. Remember, binding of two acetylcholine molecules is needed to open the receptor ion channel, allowing sodium in and potassium out, which generates an end plate potential, which when multiple occur will summate to reach threshold and lead to depolarization. Or if it doesn't reach threshold, meaning that you do get some small openings that happen but not enough to reach the threshold, then this is known as a miniature end plate potential, or in the textbooks it's shortened to a MEP. So this does not lead to a contraction, and in fact small amounts of leaked acetylcholine occur all the time, but it's only when enough is released that this causes contraction. In fact, we release about 100 to 200 vesicles, which we said earlier contain about 10,000 molecules of acetylcholine, which is actually 2 to 3 times as much as you need to actually cause an end plate potential. But it guarantees that the signal to the muscle is heard by the muscle, as it would be a catastrophe if whenever we tried to move, our muscles would only sometimes respond and other times not respond. So this excess is the concept that you'll come across known as a margin of safety at the neuromuscular junction, meaning that we over-release the amount of acetylcholine that we need to guarantee that we reach threshold when we want to move. And when we release not enough acetylcholine, which happens all the time and it's not quite reaching threshold, all this does is lead to these miniature end plate potentials, which is important that they don't cause threshold. Otherwise, we would have lots of twitching and uncontrolled movements throughout our body. So finally, acetylcholinesterase in the cleft rapidly breaks down acetylcholine into acetate and choline, terminating the signal. And choline is taken back up in the presynaptic neuron, as we mentioned before, while acetate diffuses away. So now that we understand how the neuromuscular junction creates an action potential that leads to a muscle contraction, you can actually go through these steps and then figure out how you could produce a neuromuscular block. So the 4 different ways that this can occur is you either prevent acetylcholine synthesis, you either prevent acetylcholine release, you deplete the acetylcholine stores so there's none left to release, or you block the acetylcholine receptor. And this is where people get really bogged down, I find, and they only focus on the last point because that's what we use in anesthetics. That's how our depolarizing and non-depolarizing neuromuscular blockers work. But before we talk about all of that, let's just talk about each of the other steps first, and then we'll briefly mention the depolarizing and non-depolarizing neuromuscular blockers at the end. So the first mechanism was prevent acetylcholine synthesis. So acetylcholine is made in the nerve terminal by combining choline, which is taken up by extracellular space, and acetyl-CoA, which is catalyzed by choline acetyltransferase. If you block the uptake of choline, you can't make acetylcholine essentially. And one example of this is a drug we don't use clinically, but it's called hemicholinum, which inhibits the high affinity choline transporters, which means there's less choline, less acetylcholine is made, meaning that there's less neurotransmission. As mentioned, we don't use it clinically, but it's an important experimental tool that can be used in labs, and it illustrates how cutting off the supply line of raw materials shuts down the whole process. Next is the prevention of acetylcholine release. So if acetylcholine is made, it still needs to actually be released from the cell, which is through a process called exocytosis. And this depends on calcium entry and the snare proteins that fuse the vesicles with the membrane. So toxins like botulinum toxin act here. They actually cleave the snare proteins, making them non-functioning, like synaptobrevin and SNAP25 or syntaxin, preventing vesicle fusion and acetylcholine release. So if no acetylcholine is in the cleft, then there's no muscle contraction. So this leads to what's called flaccid paralysis, which we take advantage of with Botox, which targets specific muscles for spasticity. And of course, people also use it for cosmetics to prevent wrinkling by stopping that muscle from contracting in the forehead to cause wrinkles. But probably more relevant to us is how drugs affect this. And essentially anything that antagonizes the nerve action potential getting to the cleft, such as local anesthetics, will also cause a paralysis or a motor block of that particular motor end plate of the nerve. And anything that antagonizes voltage-gated calcium channels, such as magnesium, and antibiotics like aminoglycosides, such as gentamicin, can also antagonize and stop release of acetylcholine, or at least reduce the release, which potentiates neuromuscular blockers. It's not completely clear about aminoglycosides, but it's thought that their mechanism is to do with calcium channels, because if you give them calcium chloride, it tends to reverse the effects of the aminoglycoside. So increasing the calcium levels improves it. Volatiles may also inhibit them, though usually they're quoted to antagonize L-type voltage-gated calcium channels on smooth muscle, but volatiles may also work in a similar way on these calcium channels, as they're all voltage-gated calcium channels. They may also work by simply decreasing your nervous system activity through suppression of your brain, which means that you'll have less action potentials from your motor cortex going to the muscle itself as well, which potentiates the neuromuscular blocker from that point of view. Calcium channel blocking drugs are also implicated, and furosemide as well, as it's thought to inhibit cyclic adenosine monophosphate phosphodiesterase pathways, which can reduce signaling to release calcium. A condition that sometimes comes up is Lambert-Eaton syndrome, where there are antibodies to the voltage-gated calcium channels, which leads to weakness. And usually this weakness will improve with use, as there's gradual increasing of acetylcholine being released into the synaptic cleft. So less is needed to release into the cleft to reach threshold when you're constantly trying to move that muscle. But when you stop moving that muscle, the acetylcholinesterase breaks it all back down, and you essentially have zero acetylcholine in the cleft again. So you need to rebuild it back up. And this is why their weakness tends to improve with movement. The third mechanism is depletion of acetylcholine stores. Now, some drugs don't prevent release directly. They cause excessive release, leading to depletion. There's no specific drugs that we use that use this pathway, but there are certain toxins, such as the toxin from the black widow spider venom, which also cause massive acetylcholine release, temporarily exhausting the nerve terminal. And I think a point here to also mention is that your depolarizing neuromuscular blockers, such as suxamethonium, they don't deplete acetylcholine stores, and some people get this mixed up. It's the fact that they actually bind and activate the receptor directly, but I'll talk about that later. Tetanus is sometimes brought up here as well, but tetanus toxin from Clostridium tetani works by cleaving membrane binding proteins such as SNARE, which means locally it blocks the release of acetylcholine from the motor terminals or the motor neurons, similar to botulinum toxin, though this is usually a less prominent feature of tetanus. The more important feature of tetanus is that centrally the toxin blocks release of GABA and glycine, leading to excessive and uncontrolled release of acetylcholine at peripheral neuromuscular junctions due to uninhibited motor neuron firing. This leads to no true depletion of acetylcholine. There's actually constant firing and release between brief pauses at the frequency at which the nerve fires. is what creates the summation in the muscle depending on how long the particular muscle refractory period is, which I explained in the video, which I'll link above, about how cardiac muscle isn't tendinizable, but skeletal muscle is. Lastly, we'll talk about drugs that block the acetylcholine receptor. So these are our classic neuromuscular blocking agents, and the two main types are non-depolarising blockers and depolarising blockers. So our non-depolarising blockers are things like rocuronium, vecuronium, pancuronium. They're competitive antagonists. They bind to the nicotinic receptor without activating it, preventing acetylcholine from binding. And this produces the neuromuscular blockade. While depolarising blockers like succinylcholine, also known as succinylphonium, these activate the receptors directly, causing depolarization, but they stay bound. And this is called a phase 1 block. But with prolonged exposure, the receptor becomes desensitized and you get a phase 2 block that behaves similar to a non-depolarizing block on a trainer 4 monitor. The clinical result in both cases, though, is that the muscles can't contract, leading to flaccid paralysis. And it's worth mentioning here that to achieve a block, you need to block about 70% of the nicotinic acetylcholine receptor for a non-depolarizing neuromuscular blocking drug. And this is due to that concept of the margin of safety, where we release about 2 to 3 times more acetylcholine than we need to produce synaction potential in the muscle. So to overcome this, you need to block a significant proportion of receptors. For the depolarizing neuromuscular blockers, you only need to activate about 20% to achieve a block. And it's interesting to note that succinylphonium is only broken down in the plasma and not the neuromuscular junction, as it's mostly broken down by butyrylcholinesterase and not acetylcholinesterase. And only about 10 to 15% of the dose you give actually makes it to the neuromuscular junction, as it's broken down on the way to the neuromuscular junction. And this is actually a really important concept to understand now, because the offset time for succinylphonium is dependent on the diffusion away from the neuromuscular junction to then be broken down in the periphery. And this is why people with plasma cholinesterase deficiencies or genetic mutations that prevent the breakdown have prolonged blocks, because there's decreased gradient to remove the drug from the neuromuscular junction. This concept also applies to non-depolarizing neuromuscular junctions, except for that their offset time is dependent on dissociation away from the receptor, which is dependent on a gradient or competition from acetylcholine at the receptor site to knock them off the receptor. But either way, they need to dissociate from the neuromuscular junction as well. So we've covered a lot in this video. We discussed neuromuscular junction physiology, how the neuromuscular junction works, the 4 ways a blockade can be produced. And it's really opened my mind to beyond just talking about depolarization and non-depolarizing muscle relaxants. So I hope this video has helped. If there's any questions, anything you want to add, feel free to leave it in the comments. And best of luck with exams.